Hello again. We are in document 8, Diagnostic Applications of Antibody Properties. We arrive to the second test, which is the immunomarking test, mainly the ELISA technique. Indirect ELISA technique, what is its principle? ELISA, enzyme linked immunosorbent assay, is a plate-based assay technique designed for detecting and quantifying peptides, proteins, antigens, hormones, and antibodies. In the indirect ELISA, we detect and quantify antibodies, and indirectly we diagnose the disease. So we detect the presence of antibodies in a patient's serum. If the screened antibodies are found in the patient serum, this means that this patient is infected with this disease. For example, to see whether a patient is infected with the HIV virus, we can use the ELISA technique in order to detect and quantify the anti-HIV antibodies in the serum of the patient. If the result is positive, if the serum of the patient contains anti-HIV antibodies, this means that the patient is infected with the HIV virus. So in the indirect ELISA, the antigen is coated to a multi-well plate and is detected in two stages. For example, if we diagnose the infection with the HIV virus, in the well plate, we coat the HIV antigens and we add to the coated antigen the test serum, the serum to be tested. The serum to be tested, we suppose that it might contain antibodies anti-HIV. The anti-HIV antibodies that could be found in the serum of the patient are called primary antibodies. Then an enzyme labeled secondary antibody is used in order to detect the presence of the primary antibodies. The secondary labeled antibody is specific to the primary antibody. It binds to its constant region. And if the serum contains anti-HIV antibodies, for example, this secondary antibody will bind to the anti-HIV antibodies, and the enzyme here, when we add its substrate in the final step, we obtain a colored product, a signal indicating the presence of anti-HIV antibodies in the serum and confirming the diagnosis the patient is seropositive for HIV. So what are the steps of the indirect ELISA technique? The first step, before talking about the steps, we have took the case of HIV infection. In an individual in contact with HIV, the presence of anti-HIV antibodies in his serum is proof of his infection. The patient is thus seropositive with respect to HIV. The detection of antibodies directed against the antigenic determinants or epitopes of HIV will lead to defining the seropositivity or the seronegativity of the person. Here is the structure of the HIV virus. The envelope of the HIV virus shows many glycoproteins. These glycoproteins are the antigens used in the ELISA technique. So the immune system produces antibodies specific to the glycoproteins of the HIV envelope. So the first step is the antigen coating. It consists in incubating in the wells the antigen solution, the red triangles, specific for the antibody we search for. The plates are incubated at 4 degrees Celsius overnight. The wells are then washed to remove excess antigens. So the first step is the antigen coating. We deposit the HIV antigens, for example, in the bottom of the wells. Then we wash the wells in order to eliminate excess antigens. The second step consists of adding the tested serum that could contain anti-HIV antibodies. So the second step is the fixation of the antibody to be assayed or to be tested. The antibody solution, the serum to be tested, is incubated at 37 degrees in the wells, which are coated with the HIV antigens, for approximately 30 minutes to 2 hours. The antibodies specifically bind to the antigens. If the serum of the patient contains anti-HIV antibodies, in blue here, so these antibodies will bind to the coated antigens, and after wash, the non-specific antibodies found in the serum will be eliminated, but the antibodies specific for the HIV antigens remain bound to the antigens. They will not be eliminated after wash. Only the non-specific antibodies found in the serum will be eliminated since they don't bind to the HIV antigens. The third step is the fixation or the addition of the detection antibodies, which are anti-immunoglobulin antibodies. What does it mean? The detection antibody solution is incubated at 37 degrees for approximately 30 minutes to 2 hours. The detection antibodies specifically bind to the constant region of the tested antibodies. The wells are then washed to remove excess detection antibodies with wash buffer. The detection antibodies are coupled to an enzyme. This enzyme will make a chemical reaction with a substrate and will emit a fluorescence color indicating the positive result or the infection with HIV. The secondary antibody or the detection antibody are specific for the constant region of the primary antibodies or the antibodies to be tested. If the antibodies to be tested are present in the serum of the patient, they remain bound to the HIV antigens. So when we add the secondary antibody, this secondary antibody will bind to the constant region of the blue antibodies, and thus the secondary antibody will not be eliminated by wash since it is bound to the primary antibody. So the detection antibodies are coupled to an enzyme which, in the presence of its substrate, transforms the substrate into a colored, detectable, and measurable reaction product. Note that the secondary antibody are anti-immunoglobulin. They are anti-immunoglobulin. Now, the final step, reveal of the attached antibodies. A solution containing the substrate for the enzyme is incubated at room temperature and in the dark for 10 minutes. So we add the substrate of the enzyme. If the secondary antibody is found in this solution after wash, so the enzyme will transform the substrate into a colored product. We have a yellow color that appears. This yellow color indicates that the secondary antibody is present since the enzyme was able to transform the substrate. So the secondary antibody is present. Indirectly, the primary antibody, the screened antibody, is also present. And indirectly, the patient is affected with HIV. That's why we call it indirect ELISA technique, because we indirectly diagnose HIV infection or any other infection by screening the presence of specific antibodies in the patient's serum. The intensity of the color is proportional to the quantity of the enzyme present and therefore to the concentration of antibody to be assayed. So more the color is intense, more the number of or the quantity of antibody is higher. So the higher the intensity of the color, the higher the amount of antibodies found in the serum. So the ELISA technique is a qualitative and quantitative technique. The yellow color indicates that the patient is seropositive. So we have two possible cases, seronegative case or seropositive case. If the tested serum incubated with the HIV virus does not contain anti-HIV antibodies, so the other non-specific antibodies will be eliminated after wash. So the well does not contain antibodies. So when we add the secondary antibodies which are coupled to the enzyme, they will not bind to any antibody because there is no antibodies found in this serum after wash. In turn, the secondary antibodies are also eliminated by wash since they are not bound to antibodies. So when we add the enzyme substrate, the substrate is not transformed into a colored product since the enzyme is not present. It was eliminated by wash. In this case, there is no yellow color indicating the seronegativity of the diagnosis. So the patient is not affected with HIV since its serum does not contain anti-HIV antibodies. However, here in the second case, if the serum contains anti-HIV antibodies, they bound to the coated antigen, to the adsorbed antigen. They are not eliminated after wash. So the secondary anti-IG antibody binds to the constant region of anti-HIV antibodies. They are not eliminated by wash. Now when we add the substrate, the enzyme transforms the substrate into a yellow colored product, indicating a seropositive test. So the patient has in his serum, anti-HIV antibodies, thus the patient is affected with HIV. Now we pass to the third test, which is the immunofluorescence test. It's a technique used to detect cellular or tissue antigens. As in serological tests, the antibody binds to its target antigen in a stable manner, which allows by simple washing to remove the unbound antibodies and to reveal only the specific binding of the antigen. In this technique, a fluorescent dye is attached to the specific antibody and allows direct detection of the antigen to be analyzed. So here we have the tissue that might contain the screened antigen. We can add ready-made antibodies coupled to a fluorescent substance. When the ready-made antibody is added to the tissue sample here, if the specific antigen or the corresponding antigen is present, so the antibody binds to the antigen and emits a fluorescent color indicating a positive test and the presence of the screened antigens. It's the direct immunofluorescence technique. On the other hand, we have the indirect fluorescent technique where fluorescent anti-Ig antibodies are used to detect antibodies already fixed on the tissue antigen. In this case, we talk about indirect fluorescence. Note that the fluorescence can be revealed or detected under UV fluorescence microscope. So in the indirect fluorescence technique, we use secondary antibodies coupled to a fluorescent substance. These secondary antibodies bind to the primary antibodies that are already fixed to the antigen or to the tissue antigen. In this way, we detect the antibodies and indirectly we detect the infection. These are the three tests used in laboratory medicine and they show the specificity of antibodies to their antigens.